Carbohydrate metabolism, Krebs cycle. After the conversion of pyruvate to acetyl-CoA through the pyruvate dehydrogenase complex, the acetyl-CoA becomes oxidized through the series of steps of the Krebs cycle. It is important to note that two acetyl-CoA are formed from two pyruvate molecules when oxidizing a molecule of glucose. We will now review the steps of the Krebs cycle, outlining key portions of the process. As with glycolysis, we will not go over each step, enzyme, and intermediate, but instead we will emphasize key reactions of the process involved in the formation of key products or involved in the regulation of the Krebs cycle. As an overall summary, the Krebs cycle begins with a molecule of acetyl-CoA joining with oxaloacetate, which forms citrate. Citrate then goes through a series of steps, eventually reforming oxaloacetate. In the process, key compounds including NADH, FADH2, ATP, and CO2 are formed. Here is a diagram of the Krebs cycle. We will go through the cycle highlighting key steps of the process. As mentioned, the first step involves the 2-carbon compound acetyl-CoA joining with the 4-carbon compound oxaloacetate. So here's the 2-carbon acetyl-CoA joining with the 4-carbon oxaloacetate to form the 6-carbon compound citrate. From there, citrate forms isocitrate. We will discuss in more detail the next two steps, which both involve the release of a carbon as CO2 and the formation of NADH. The first of these two steps involves the conversion of isocitrate to alpha-ketoglutarate. You see here that we release a carbon as CO2 and we form an NADH from the oxidized NAD. Therefore, you can see the 6-carbon isocitrate then goes to the 5-carbon alpha-ketoglutarate. This next step also loses a carbon as CO2 and also forms NADH. And this is the step of alpha-ketoglutarate being converted to succinyl-CoA. Interestingly, this reaction of alpha-ketoglutarate to succinyl-CoA is called the alpha-ketoglutarate dehydrogenase complex, and it has many similarities with the pyruvate dehydrogenase complex. You can see, like the pyruvate dehydrogenase complex, this step converting alpha-ketoglutarate to succinyl-CoA also utilizes a molecule of coenzyme A. Additional similarities is the release of CO2 and the formation of NADH. Another important point here is you see that the 5-carbon alpha-ketoglutarate with the release of carbon as CO2 is then formed to the 4-carbon compound succinyl-CoA. No other carbons are lost through the process, which then continues on to eventually form the 4-carbon oxaloacetate. The next key step we will emphasize is the conversion of succinyl-CoA to succinate. During this step, a GTP or an ATP molecule is formed. The next step, which involves the conversion of succinate to fumarate, involves the formation of FADH2 from the oxidized molecule FAD. The last step we will discuss is the conversion of malate to oxaloacetate. Here, another product is formed, where you see that the oxidized NAD is converted to the reduced NADH. You can see that we have focused on the steps that involve the formation of either ATP or the formation of the reduced NADH or the reduced FADH2. These steps are important to note because the NADH and FADH2 molecules will then go on to the electron transport chain to eventually produce ATP. Keeping track of these products will be important when calculating the ATP yielded during the oxidation of one molecule of glucose. Therefore, one turn of the Krebs cycle specifically forms 3 NADH, and you can see here one NADH is formed when converting isocitrate to alpha-ketoglutarate. One is formed in the conversion of alpha-ketoglutarate to succinyl-CoA. And then last we have formation of NADH in the conversion of malate to oxaloacetate. So we have the formation of 3 NADH, 1 FADH2. Again, you can see here that's in the conversion of succinate to fumarate. We have the formation of an ATP or GTP molecule, again in the conversion of succinyl-CoA to succinate. And then also we have the release of CO2 in the 2 initial steps where NADH is formed. Here, this diagram also outlines the steps of the Krebs cycle, though it additionally includes the enzymes catalyzing each step. While you will not be tested on each enzyme of the process, it will be important to know the enzymes involved in the regulation of the Krebs cycle, which we show in this diagram. This slide outlines the enzymes involved in the regulation of the Krebs cycle, as well as the enzyme catalyzing the pyruvate dehydrogenase complex. The enzyme regulators of the Krebs cycle include citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase. Here in this diagram, you see the steps of the pyruvate dehydrogenase and the Krebs cycle. In red, you see the metabolic signals either inhibiting these key enzymes or activating these key enzymes in green. First, we'll look at the pyruvate dehydrogenase complex, which is the conversion of pyruvate to acetyl-CoA. You see here that both acetyl-CoA and NADH are inhibitors of pyruvate dehydrogenase, whereas calcium activates its activity. Next, for citrate synthase, we see that citrate and succinyl-CoA, you see the arrow going from succinyl-CoA here, both citrate and succinyl-CoA inhibit the activity of citrate synthase. Next, for isocitrate dehydrogenase, we see that calcium and ADP activates its activity, whereas NADH is an inhibitor. Lastly, for alpha-ketoglutarate dehydrogenase, calcium activates its activity, whereas NADH and succinyl-CoA inhibits. Therefore, it will be important to know each of these enzymes regulating the pyruvate dehydrogenase complex and the Krebs cycle. Also, it will be important to know the metabolic signals that increase or decrease their activity. A pattern you may recognize is that of product inhibition, where you see that the immediate or eventual products of reactions inhibit the enzyme used in their formation. For example, you see that acetyl-CoA inhibits pyruvate dehydrogenase. Also, you see that calcium promotes the activity of several of these enzymes. In the muscle cell, calcium is released from the sarcoplasmic reticulum during muscle contraction. Therefore, in this case, calcium is acting as a signal, indicating muscle contraction and an increased need to produce ATP.